Imagine inheriting 4 exact, identical copies of the largest, most dense encyclopedia in the world.
Right, which sounds great at first.
Yeah, exactly. At first you might think, well, great, more information. But when you actually try to use it, the sheer volume is just, it's visceral, paralyzing chaos.
Oh, absolutely.
Every time you try to look up a simple recipe, the pages are stuck together, the indexes are, randomly cross-referencing each other, and your brain just blue screens. You just can't process it.
And the kicker is, you can't just throw 3 of those copies away. The machinery of your cell forces you to read all 4 encyclopedias at the exact same time.
That sounds exhausting.
It really is. It's this sudden, massive influx of genetic data. You certainly have all the raw material to build something incredible, but without a biological filing system, the cell's basic operating system just completely jams up.
Okay, let's unpack this, because today's deep dive is all about how nature creates that filing system out of thin air.
Yes, it is.
We've got our hands on a really fascinating 2026 paper published in the journal Nature. The researchers mapped the genomes of snow carps. And if you aren't familiar, these are this ridiculously tough group of fish living in the extreme, freezing environments of the Qinghai Tibetan Plateau.
Right. We are talking high altitude, very low oxygen, freezing glacial rivers. It's an incredibly harsh environment. And, it sets the stage for a remarkable study.
It really does.
For over 50 years, biologists have known that the sheer complexity of vertebrates the evolution of jaws, limbs, and eventually humans was driven by these ancient chaotic events where entire genomes duplicated themselves.
Right.
But how a wildly unstable duplicated genome eventually calms down into a normal paired state, well, that has been a total biological mystery.
And they call that stabilization process rediploidization.
Exactly, rediploidization.
So our mission today is to explore the mechanical trigger that solves this ancient puzzle. We're looking at how these high-altitude fish are actively organizing their duplicated encyclopedias right now in real time.
Which is just incredible to witness.
But before we get to the fix, we really need to understand the disaster itself. I mean, what causes an entire genome to double in the first place?
So there are 2 main ways a whole genome duplication happens. The first is when 2 completely different species hybridize.
Like a crossbreed.
Right. Think of it as 2 different companies deciding to merge. They have different corporate cultures, different rule books, and they kind of clash until they figure out a completely new hierarchy.
Okay, yeah, that makes sense. A merger of 2 distinct entities.
But the snow carps went through something much stranger, something called autopolyploidy.
Autopolyploidy?
Yeah. This is when a single species duplicates its own completely identical chromosomes.
Wait, really?
Yes. So going back to our corporate analogy, imagine a company just perfectly cloning itself overnight.
Oh, wow.
You walk in on Monday morning, and there are 2 identical accounting departments, 2 identical HR departments, sitting at the exact same desks, trying to do the exact same jobs.
That sounds like an absolute nightmare. I mean, you'd have 4 people fighting over the same keyboard. Why would a fish's biology subject itself to that level of inefficiency?
Well, if we connect this to the bigger picture, it usually comes down to survival. The origin of the snow carp group dates back roughly 10 to 30 million years ago.
Okay.
And that timeline coincides perfectly with massive climatic upheavals and the intense tectonic uplift that actually created the Qinghai Tibetan Plateau.
So the environment was changing rapidly.
Violently changing. Normal, slow evolution just wasn't going to cut it. Duplicating their entire genome provided this instant, massive reservoir of raw genetic material that they could mutate to adapt to the freezing water.
Okay, but I mean, it's not like we have fossilized DNA from a frozen river 30 million years ago. How do researchers look at a modern fish and know it cloned its own genome in the distant past?
They use a really clever counting technique called Kamer frequency distribution. Yeah. Basically, if you chop a genome up into tiny, unique genetic words, you can run a computer program to count how many times each specific word appears.
Oh, okay.
In a normal genome like ours, most unique words appear twice, one from the mother, one from the father. But when they ran this on the snow carps, they saw massive, undeniable spikes where unique words appeared 4 distinct times.
Meaning they're carrying 4 copies of everything, a tetraploid genome.
Exactly. For most of them, yes. But, nature always pushes the limits. They found one species that has 6 copies.
6?
Yeah, hexaploid. And one truly baffling species, a fish called Platypterodon dipagon, has 20 copies of its genetic material.
20 copies? Wait. If my cells had to juggle 20 copies of every single biological instruction just to keep my heart beating, the energy waste alone would kill me.
It's highly inefficient, yeah.
How does a cell physically survive that? I mean, when a cell divides, chromosomes have to pair up. If you have 4 or 20 identical twins in the room, how do they know who to pair with?
Well, that's the thing. They don't. And that is the lethal danger of cloning your own genome. When you have 4 identical chromosomes, they pair up completely randomly during reproduction. It is basically genomic roulette.
So it's just chaos.
Total chaos. This is incredibly unstable and leads to massive genetic errors. To actually use the duplicated genes to evolve, the genome absolutely must undergo rediploidization.
It has to force those 4 random players to settle down into 2 strict, stable pairs, just like normal chromosomes.
Precisely.
Okay, so just to ground this for a second, let's sort of quick recap. Think about your own body right now. Think about the sheer biological energy required to simply exist. If your cells suddenly had 4 active copies of every gene, the structural chaos would be devastating.
Definitely.
But at the same time, if you could stabilize it, you'd have an entire backup set of genes that you could mutate to, say, breathe underwater or survive sub-zero temperatures. So I have a question for you listening. If your cells suddenly had 4 copies of every single gene, which ones would you want to mutate and take on new superpowers?
That's a great question to think about, because that's the exact tightrope the snow carp is walking.
Right, which brings us to the actual physical chromosomes. The researchers know the fish has a 4-copy tetraploid genome, but when they looked through the microscope to physically count the chromosomes in one specific species, Schizothoracinae young husbandi, the math completely fell apart.
It was a glaring discrepancy. For this specific fish, the base set of chromosomes, what we call the haploid number, is 25.
Okay, 25.
Therefore, if it has 4 copies of everything, a simple calculation tells us there should be exactly 100 chromosomes floating in the cell.
4 times 25. Simple math. But they didn't find 100.
No, they only found 90.
Wait, did the fish just shed the extra weight? I mean, 10 chromosomes is a lot of genetic information to just misplace.
Losing them was the logical first guess, for sure. But when the researchers used a technique called genomic synteny, which is comparing the physical map of the snow carp's chromosomes to other related fish, they realized nothing was actually missing.
So where did they go?
The chromosomes had actually fused together. 2 individual chromosomes physically connected end to end to become one giant chromosome.
Wow.
And they found 5 distinct pairs of these fusions, which perfectly accounts for the drop from 190.
So they just glue themselves together. But we have 4 identical copies of every chromosome in this fish. Are all 4 copies fusing into some kind of massive superchromosome?
Well, no. And this is the critical discovery of the whole paper. The fusions were unbalanced.
Unbalanced?
Yeah. Out of the 4 identical copies, let's call them copies 1, 2, 3, and 4, only 2 of them fused together. The other 2 identical copies remained completely separate, entirely unfused.
Here's where it gets really interesting. Let me try to picture this. It's like a 4-legged race. You have 4 runners on a field representing our 4 identical chromosomes.
Right. Good analogy.
Normally, they can link arms with whoever they want, which creates that dangerous, chaotic roulette we talked about.
Yeah.
But if you suddenly take 2 of those runners and tie their shoelaces together, creating a fused pair, they are physically tethered.
Exactly.
But wait, if you tie 2 chromosomes together, wouldn't that just make the chaos worse? Now you have a tangled knot trying to interact with 2 free runners.
You'd think so, but the fusion actually changes the physical rules of the game. Yeah. Because those 2 chromosomes are glued end to end, the resulting joined pair is physically twice as long.
Oh, I see.
It has a completely different shape. The cellular machinery that pairs chromosomes together during reproduction is incredibly sensitive to physical shape. It basically can't align a massive, long, fused chromosome with a short, unfused one. It's like trying to fit a square peg into a round hole.
Because the shape is so wildly different, they physically cannot pair with the unfused ones anymore.
Precisely. They are forced to find their matching shape. The fused ones are forced to pair only with the other fused ones, and the unfused are left to pair only with the unfused.
Wow.
This unbalanced structural anomaly is the exact physical anchor that stops the chaotic roulette. It forces the 4-way free-for-all to segregate into 2 separate, stable, 2-way pairings.
So a random physical mutation, literally just 2 chromosomes bumping into each other and sticking accidentally, solves the biggest crisis of the duplicated genome.
Yes, exactly.
It acts as an anchor.
Yeah.
But once that anchor drops, does the whole genome instantly snap to attention and stabilize?
It is the exact opposite of instant. The researchers discovered that stabilization is an incredibly slow, agonizingly gradual geographic crawl.
A geographic crawl.
Yeah. They proved this by looking at something called case values, which act like a molecular clock.
Okay, wait, how do you read a clock on a chromosome?
So when DNA copies itself over millions of years, it inevitably makes tiny, harmless typos. We call these synonymous substitution.
Harmless typos, got it.
Because they don't harm the fish, these typos accumulate at a very steady, predictable rate. By counting the number of these typos between two chromosomes, you can calculate exactly how long ago they stopped randomly mixing and started evolving independently.
Oh, so the more typos they have in common, the more recently they separated. And the more different their typos are, the older the separation.
Exactly. And when they mapped these typos across the snow carp genome, they found that the stabilization happened in 3 distinct historical waves.
Over what kind of timeline? Were we talking hundreds of years, thousands?
Oh, much longer. The oldest wave, which was just wave 1, started roughly 30 million years ago.
Wow, 30 million.
Right around the time of the original genetic duplication. The second wave didn't happen until 10 to 20 million years ago. And the third wave is so recent that the molecular clock has barely ticked.
That is just wild.
Yeah.
Different parts of the exact same genome are stabilizing tens of millions of years apart.
It's a very slow process.
But you also called it a geographic crawl. What does the geography of a chromosome have to do with it?
Well, when the researchers looked at a single individual chromosome, they found the stabilization doesn't happen all at once along its length. It starts precisely at the physical site where the two chromosomes fuse together.
Let me try to visualize that. Imagine taking a long, shallow trough of water and dropping a single bead of red food coloring right in the center. The fusion site is where the drop hits. And the stability, the rediploidization, starts exactly at that anchor point and slowly, over millions of years, bleeds outward toward the distant ends of the chromosome.
But that visual is actually perfect. The data supports that entirely. The centers of the chromosomes, right near the fusion, have a huge number of independent typos. They're highly stable and paired up nicely.
Right.
But the distant ends, the arms of the chromosome, they're still wildly chaotic. They're still randomly mixing all 4 copies together.
Wait, my brain is having a hard time with the physical reality of this.
Yeah.
You're saying a single physical strand of DNA inside a living fish right now is half stable in the middle and a chaotic 4-way roulette game on the ends.
Yes.
How does a cell even divide without tearing that hybrid structure completely apart?
It is a violent, tangled process, and the researchers actually photographed it.
They took a picture of it.
Yeah, using a technique called FESH-ish imaging, fluorescence in situ hybridization.
Okay, FESH.
Essentially, they engineered fluorescent chemical tags that only bind to specific DNA sequences. So they literally paint the chromosomes so they glow under a microscope.
Well, that's cool.
And when they watched the cells attempt to divide, what's fascinating here is that they saw literal visual proof of this hybrid state frozen in action.
What does a half-stable, half-chaotic chromosome even look like glowing under a microscope?
Normally, dividing chromosomes look like simple, neat little rods or, X shapes. But here, the researchers saw massive, glowing, ring-like and chain-like structures.
Rings.
Yeah, they saw huge chains of 6 chromosomes linked together, forming complex, tangled geometries. Because the centers of the chromosomes are desperately trying to pair strictly in 2s, but the arms are reaching out and grabbing anyone they can, so they form these bizarre, tortured shapes.
That sounds like a biological car crash frozen in time.
It really is evolution caught in the act. These glowing rings prove that regaining stability isn't a switch that flips. It is a brutal physical geography that has to be slowly conquered over tens of millions of years.
Okay, let's take a breath and gather all these threads for a quick recap.
Good idea.
We've learned that doubling your genome gives you the fuel to survive extreme environments. But it creates a chaotic roulette where chromosomes pair randomly.
Right.
The return to a stable paired genome is triggered by unbalanced fusions, where the new long-shaped force is matching. And this stability isn't instant. It bleeds outward from the fusion point in historical waves that take millions of years, creating bizarre, tangled, glowing rings in the process.
That is the mechanical reality of the snow carp's genome, yes.
So, to you listening, what stands out to you? Do you think having a hybrid chromosome gives the snow carp an immediate evolutionary advantage? Or is it just a messy, necessary transition phase to survive the duplication?
It's definitely a messy transition.
So think about what it means to be in that phase. The fish is swimming around in glacial water with a genome that is literally tangled in knots. But the big question remains, while the chromosome is doing this million-year geographic crawl, what is happening to the actual genes? I mean, we duplicated the whole encyclopedia. Are the extra genes just sitting there doing nothing?
And this is where the story shifts from physical structure to actual function. Once the chromosomes are forced into these separate pairs, the fused ones over here, the unfused over there, the duplicated genes on them are finally free to evolve independently.
Okay.
We call these duplicate genes analogs.
Analogs. And what is the ultimate fate of these analogs?
Well, the study revealed a highly asymmetric, almost tragic relationship between the identical copies. It is not an equal partnership at all.
Tragic how?
First, the researchers looked at gene loss. Maintaining extra genes requires a lot of cellular energy. Over millions of years, evolution tends to aggressively delete redundant genes just to save fuel.
Makes sense.
They found that this dilution heavily targets the fused chromosomes. In fact, over 71% of all the deleted duplicate genes were ripped from the fused copies.
So what does this all mean? If I bring back our corporate merger analogy, we perfectly cloned the company. Now, the unfused identical department becomes the main headquarters.
Yeah.
It's protected.
Exactly.
Meanwhile, the fused department gets mercilessly downsized and stripped for parts, maybe pushed into weird niche projects. But wait, why? They started as perfect identical copies. Why does the cell treat the fused ones like second-class citizens?
It really comes back to the violence of the fusion itself. When two chromosomes slam together end-to-end to create that anchor, the impact zone isn't clean. The regulatory regions, the genetic on and off switches that sit around the genes, can get severely damaged or buried deep within the new folded structure.
Oh, so the instructions are still there, but the cell can't reach the light switch to turn them on.
Exactly. Because the cell's machinery finds it harder to read the fused genes, it naturally defaults to the pristine, untouched, unfused copies.
That's rough.
The researchers proved this by analyzing RNA sequencing across 11 different tissues in the fish, like the gills, the brain, the liver.
And RNA tells us how actively a gene is working.
Right. They found that the genes on the unfused chromosomes are doing significantly more heavy lifting. About 20% of the gene pairs are unfused biased, meaning the unfused copy is running the show.
Wow.
Only 10% are fused biased.
The unfused chromosomes are undeniably the boss. But does that mean they've completely split their responsibilities? If you have two surviving accounting departments, maybe one handles daily taxes, and the mutant one figures out how to survive freezing water, did the fish evolve totally new superpowers yet?
Well, this raises an important question, right? Have they separated their functions? That was the final test. The researchers took juvenile snow carps and exposed them to 24 hours of severe cold stress, dropping the water down to a brutal 5 degrees Celsius.
Oh, wow. 5 degrees.
They wanted to see if the fused and unfused genes would react differently in the gills and the heart. Would one handle the baseline metabolism while the other fought the cold?
Did they divide the labor?
Surprisingly, no. They moved in complete lockstep. If the unfused gene ramped up its activity to fight the freezing water, the fused gene ramped up right alongside it. If one shut down, the other shut down.
So they don't have different jobs yet. They're doing the exact same thing, just one is a bit louder than the other.
Yes. And this provides a profound insight. It tells us we are observing the absolute earliest stages of evolution following a duplication. The genes have started to separate in their power levels, but they haven't yet mutated into entirely new roles. Biologists call this subgenome dominance.
Subgenome dominance. That implies one genome is asserting power over the other.
Normally, we only see subgenome dominance when two entirely different species hybridize. The stronger species genome suppresses the weaker one.
Right, the merger clash.
But the snow carp duplicated its own identical genome. The dominance we are seeing here is driven entirely by internal architecture. Just because two identical chromosomes physically fused together to save the cell from chaos, they sacrifice their own gene's ability to be read. The physical structure alone dictated their evolutionary doom.
The very anchor that saved the genome doomed the genes that formed it. That is a brilliant, almost Shakespearean mechanism.
It really is.
Let's pull all the way out here. We've journeyed from the high-altitude, low-oxygen rivers of the Qinghai Tibetan Plateau down to the tangled, glowing rings of dividing cells.
Yeah.
We've seen that when nature makes a massive evolutionary leap by doubling the entire genetic code, the resulting chaos is lethal. It requires mechanical anchors, physical fusions that change the shape of the chromosome to force order.
Exactly.
And that order doesn't happen overnight. It ripples outward in geographic waves over tens of millions of years, creating a dominant and a submissive genome out of perfect identical twins.
It is a beautiful demonstration of how biology operates. It isn't always clean or efficient. It's often about finding a messy, structural workaround, like tying two shoelaces together just to survive the immediate crisis, and letting evolution sort out the details over the next few million years.
There is a surprisingly comforting real-world takeaway here, too.
Oh, yeah.
Yeah. The next time you feel completely overwhelmed by a massive influx of information, or you're staring down a chaotic, impossible project at work, think about the snow carp. You don't have to organize the entire encyclopedia at once. You don't have to fix the whole chaotic system in a day.
That's true.
Just find two things. Find two things to fuse together. Create a single solid anchor and let the stability ripple outwards at its own pace.
It's a very practical way to view a biological crisis.
But I want to leave you with one final, slightly unsettling thought to ponder. We've been talking about these fish as if they are some distant, bizarre anomaly. But the entire vertebrate family tree is built on whole genome duplication.
Oh, absolutely.
Our own human ancestors underwent this exact same chaotic doubling hundreds of millions of years ago. We assume our genomes are perfectly stable and finished. But what if our own rediploidization isn't completely done? What if there are ancient, untamed edges of our human chromosomes that are still, on some microscopic level, echoing that chaotic four-way roulette? Are we entirely finished evolving? Or is the wave of stability still crawling across our own DNA? Something for you to chew on until our next deep dive.